Hello to all the viewers. Today I have the pleasure to talk to Professor Alexander Kai Buell from Danish Technical University, and hear his opinion on using FIDA for research on liquid-liquid phase separation. Hello, Alexander. Would you like to tell us more about yourself and your laboratory?
Hi, Maya. Of course, it's a pleasure. Yes, so my name is Alexander Buell, and I'm a professor of protein biophysics here at DTU, the Danish Technical University in Lyngby. And I'm running both the protein biophysics research group as well as a protein biophysics core facility. And so in my research group, we're mostly interested in phenomena such as protein aggregation, amyloid fibril formation, loss of solubility of proteins. You could say, whenever proteins sort of misbehave and lose their natural function because they are aggregating, forming clumps, and so on. And originally, we were mostly focusing on these phenomena in the context of neurodegeneration, notably Parkinson's disease. We worked a lot on the molecular mechanisms of that. And more recently, we've also focused some of our efforts on liquid phase separation, the topic of this presentation or this interview, whereby a protein solution spontaneously separates into dense droplets or dense part and a very much more dilute part. And there is lots of evidence that this type of phenomenon plays a role in biology, both in function and disease. And then also, since joining DTU, I've started to extend also the research into protein materials, but also food-related topics. So protein aggregation plays a role in all these different domains.
Mm-hmm. Thank you. And could you share with us what questions or challenges are currently being addressed in the field of liquid-liquid phase separation? And also, of course, how does FIDA address these?
Yes, so I mean, obviously there is many different challenges also related to the physiological effects of these. But speaking about the biophysics of phase separation, protein phase separation in vitro experiments, I mean, one part would be quantifying the thermodynamic driving forces responsible for phase separation. And another part would be to look at how compounds of various kinds can modify, can enhance or inhibit phase separation. And notably, in both of those areas, methods that allow to measure samples at high throughput using very low amounts of sample are in high demand or are quite valuable because various companies are trying to find inhibitors of phase separation because of the disease relatedness, but also in our specific case, we're interested in studying many different sequence variants, so making changes in the amino acid sequence and seeing how that affects the thermodynamics of phase separation. And for that, we would need methods that allow to get these parameters in a high throughput way using low amounts of sample. And this is really where FIDA comes to the strength or comes to our help.
Mm-hmm. And are there any other capacities that are currently missing in LLPS research? And can FIDA address them somehow?
Yes, so talking about screening of various conditions, solution conditions, inhibitors, temperature, and things like that. If you want to do that at really high throughput, there's some other methods available. For example, some microfluidic, microdroplet-based methods whereby protein solutions are encapsulated into tiny microcompartments. And some of these methods are extremely powerful and really quite wonderful, but they're also very difficult to use. So having a method available that allows to measure at high throughput, but at the same time being based on a commercially available platform that doesn't need years of training, I think is really a valuable point or a valuable addition. And so I believe actually that FIDA is sort of filling that gap or is providing this capability because the instrument is more or less turnkey or readily available commercially, and with relatively short training, implementing the methods that we're developing, a user can actually quantify the thermodynamics of phase separation and perform screens that are quite high throughput, comparable to the sort of methods that are around or better, but not needing so much expertise and so many years of training.
Mm-hmm. And since we're in this topic, could you tell us more about how does FIDA fit into the challenges at hand in the light of other technologies that are in use?
So, of course, in the field of phase separation and aggregation and so on, other methods are also being routinely used. For example, light scattering methods that would allow you to detect the formation of protein condensates or aggregates quite sensitively. Of course, other ways to get to the thermodynamics of phase separation, such as, for example, quantifying the concentrations of protein in the dense and dilute phase, can be achieved by relatively simple means, for example, by centrifuging the sample and then separating dense and dilute phase and quantifying their concentrations separately. So FIDA is complementary and kind of useful in that space because it uses less sample, it's more easily automatable. But of course, the other methods are also useful sometimes. For example, when we want to pick up really small concentrations of aggregates or traces of condensates, nanoclusters we have been studying also, that light scattering can be quite useful because it's very sensitive to the presence of small aggregated species or sort of cluster-like species. But the capability of using just tens of nanoliters of sample and in an automated way screening tens or hundreds of conditions, that's quite unique to FIDA, and we don't have any other method in our lab that would be able to do that.
Mm-hmm. And would you like to tell us more about a specific research you did?
In that area, yes. So, for example, one of the things we're quite proud about is a paper that we published this year in the journal Angewandte Chemie, international edition, where we demonstrate that phase separation, protein phase separation, can be induced inside the FIDA instrument by for example, injecting the protein together with a phase separation repressing component, which then gets removed due to the Taylor dispersion inside the capillary, and then phase separation gets induced, and then one can use that to further look at the influence of other components on phase separation. So one can add inhibitors or enhancers, and the whole thing would use only something like 50 nanoliters of sample per data point. And so that's really nice. I think we'll find great use in both academic and industrial settings. We're already talking to various people that are interested in implementing this. And another thing that I would maybe recommend people having a look at is we, together with my postdoc Shumik Ray, we wrote a review recently on different experimentally available methods to quantify the thermodynamics of liquid phase separation.
Mm-hmm.
So we did a very comprehensive literature review, also including some very recent work that was only preprint. And we also discuss our own method development on the FIDA platform in that context and compare it to some of the other available things.
Wonderful. Our viewers can actually find both papers on the literature section on our website. So I recommend that everybody can go there now and check it out. And my last question is, if you would like to share it with us, of course, what plans do you have for applying FIDA technology moving forward?
So in addition to measuring binding affinities and so on, which we're doing quite routinely with the FIDA as well, and the work we're doing on the phase separation that I just described, we're now more recently interested in looking at micellar species. So we're having various projects in a food protein-related space as well, and one of the types of proteins that we're interested in is caseins in milk, and they're of course known to form micelles of various sizes, and they complex calcium and so on. And we found out that FIDA is a really useful technique to not only quantify the formation of micelles by these proteins, but also looking at the dynamics. So how stable are these micelles upon changes in condition, upon removal of the calcium and things like that. So it turns out we need to do a little bit of development of the theory and also perform some simulations to understand the behavior of these micelles better inside the FIDA, but it's a very powerful method. It appears to quantify this, which is not otherwise so easy to quantify.
Wonderful. Good luck with that research. And thank you so much for all the insights. It's been a pleasure to talk to you.
Thank you very much.